Hi all and welcome to the Gemophile. Imagine you're working in a clinical microbiology lab and a sample arrives from a patient with a suspected bacterial infection. Before you can identify the species, before you choose the right antibiotic, you need to answer a more basic question. What kind of bacteria are you dealing with? One of the first tools you would reach for is the Gram stain, a technique developed by the Danish physician Hans Christian Gram in 1884 and still used in laboratories around the world today. In this video, we will explore the science behind why the Gram stain works, walk through the procedure step by step, and discuss how to avoid the most common errors. To understand the Gram stain, you need to know about the bacterial cell wall, specifically the molecule called Peptidoglycan. Gram-positive bacteria, such as Staphylococcus aureus, have a thick peptidoglycan cell wall directly outside their plasma membrane. Gram-negative bacteria, such as Escherichia coli or E. coli, have a much thinner peptidoglycan layer sandwiched between an inner plasma membrane and an outer membrane. This structural difference is the key to the entire procedure. When we apply the primary stain, crystal violet, followed by iodine as a mordant, a crystal violet iodine complex forms inside the cells. In gram-positive bacteria, the thick peptidoglycan wall traps this complex when ethanol is applied, retaining the purple color of the crystal violet. In gram-negative bacteria, the outer membrane is disrupted by ethanol, allowing the complex to wash out. These cells then pick up the pink counterstain safranin. Before we begin, let us go through what you will need. For equipment, you will need a Bunsen burner, an inoculating loop, microscope slides, a staining tray, and a light microscope. For cultures, you can use S. aureus as your gram-positive reference strain and E. coli as your gram-negative reference strain. For reagents and materials, you will need crystal violet stain, Gram's iodine, ethanol as your decolorizer, safranin as your counter stain, distilled water, and blotting paper for drying the slide. Before starting, put on your lab coat and gloves. Clean your work surface to reduce the risk of contamination. You will be working with live bacteria and open flames, so follow your laboratory's biosafety procedures throughout. These organisms are handled under biosafety levels 1 to 2 conditions, depending on your institution's classification. Step 1. Prepare the smear. Sterilize your inoculating loop by holding it in the Bunsen burner flame until the wire glows red hot. Then allow it to cool for a few seconds. Transfer a small amount of your bacterial culture to a clean microscope slide using a circular spreading motion to create a thin, even smear roughly the size of a small coin. Allow the smear to air dry near the Bunsen burner. Step 2. Heat fixation. Pass the slide quickly through the Bunsen burner flame 2 to 3 times, smear side up. This heat fixes the cells to the glass surface so they do not wash off during staining. Step 3. Apply the primary stain, crystal violet. Place the slide on your staining tray. Flood it with crystal violet and leave it for 1 minute. At this stage, both gram-positive and gram-negative cells are stained purple. Step 4. Rinse with distilled water. Tilt the slide and gently rinse off the crystal violet with distilled water. Be careful not to disturb the smear. Step 5. Apply the mordant, iodine. Flood the slide with Gram's iodine and leave it for 1 minute. The iodine forms a complex with crystal violet inside the cells, setting the stain. Both cell types remain purple. Step 6. Rinse with distilled water. Rinse off the iodine gently with distilled water. Step 7. Decolorization with ethanol. With the slide still tilted, flood it with ethanol for 2 to 5 seconds, then immediately rinse with distilled water. This is the most critical and most error-prone step. Ethanol disrupts the outer membrane of gram-negative bacteria, releasing the crystal violet iodine complex. Gram-positive bacteria retain this complex. Over-decolorization can cause even gram-positive cells to lose the stain, giving a false gram-negative result. Step 8. Apply the counterstain safranin. Flood the slide with safranin and leave it for 30 seconds. Then rinse with distilled water. Safranin stains the now colorless gram-negative cells pink. Gram-positive cells, which retained the crystal violet, remain purple and are only minimally affected by safranin. Step 9. Blot, dry, and examine. Gently blot the slide dry with blotting paper and examine it under the light microscope. A successful gram stain will show S. aureus as purple, which means it is gram-positive, and E. coli as pink, which means it is gram-negative. The gram stain is reliable, but small mistakes can produce misleading results. Over-decolorization is the most common error. Leaving ethanol on the slide for too long removes the crystal violet from gram-positive cells, causing them to appear gram-negative. In contrast, under-decolorization results in gram-negative cells retaining more of the stain than expected, and they may appear gram-positive. An old or degenerating culture can also give unreliable results. Very old gram-positive cells may lose the ability to retain crystal violet. This is sometimes called gram variability. Finally, a smear that is too thick prevents reagents from penetrating and rinsing evenly, making interpretation difficult. The gram stain is used across clinical, research, and food safety microbiology. In a clinical setting, it is one of the first tests performed on a patient sample to guide antibiotic selection before culture results are available, since gram-positive and gram-negative bacteria typically respond differently to different classes of antibiotics. For example, in South African tertiary hospitals, a rapid gram stain of cerebrospinal fluid is standard workflow to immediately differentiate gram-positive from gram-negative bacterial meningitis, saving critical time before definitive cultures grow. Let us pull it all together. The gram stain separates bacteria into two groups based on cell wall structure. Gram-positive bacteria with their thick peptidoglycan wall retain crystal violet and stain purple. Gram-negative bacteria has a thin peptidoglycan layer and an outer membrane. They lose crystal violet during decolorization and pick up safranin instead, staining pink. Whether you are heading into a practical class, interpreting results in a research lab, or studying for an exam, understanding the gram stain is a foundational skill in microbiology. To learn more about gram staining, check out the references linked below. That is all for now. Join us next time on The Gemophile as we explore the fascinating world of microbes together.